Right. Okay, hi guys, this video is going to be on membranes, their structure and function. So the idea of a membrane is to create compartments within a cell. So sometimes students get confused with the idea that a plasma membrane is just on the exterior of a cell. But of course inside a cell we have organelles which are membrane bound, a nucleus, mitochondria, chloroplasts if we're a plant, etc. So these all have their own individual membranes around the outside, which we would class as a plasma membrane, and that creates a compartment, a difference between the environment within the membrane and outside the membrane. The plasma membrane, the cell membrane, refers to the membrane around the outside. The membrane's function, the cell membrane's function, is to control the movement of products, reactants, things inside and outside of the cell. So this controls what moves in and out of the cell. It is not for structure like the cell wall in plant cells and in bacterial cells. It's purely to control what comes in and out. Okay? So what we need to have an awareness of is how the structure of this membrane relates to being able to carry out this function. So we say the membrane has a fluid mosaic structure. So if you think of a mosaic like tiles in your bathroom, we kind of think of like lots of pieces all kind of tiled together with kind of spaces in between, and that's a bit like the membrane. So what happens is we have a sort of basic structure, the sort of grout in between the tiles, if you like, of phospholipids. We'll talk about what those are in a second. And each phospholipid is made up of a phosphate head and fatty acid tails, along with a glycerol, but you cover more about that when we look at lipids and the difference there. So a phosphate head, change the colour here, at the surface, and the tails, the fatty acid tails, are beneath them on the interior, the hydrophobic region within the middle of the bilayer. So the hydrophobic region, when we have a phobia or we are phobic to something, that means we are afraid of it. So hydrophobic means there is no water. The tails do not like water. Hydrophobic region on the inside. The heads, we say, are hydro, meaning water, hydrophilic, which means they don't mind water, okay? So the arrangement of the phospholipids forms something called a bilayer. Bi meaning two, here's the first layer, here's the second layer. So this creates a region within the center of the bilayer which is hydrophobic, which will become important later. So if we dropped phospholipids into water, it forms something called a mycele, which would look like this. So this would give us the same arrangement, we just have a single layer rather than a bilayer here. But because of the properties of the phospholipids, this is what we get. So in the membrane, we have this bilayer here. I'll draw the phospholipids out really quick like this. And then in between each of the layers, we have dotted membrane proteins. So some of these proteins might span both layers, like this one here. We say those are proteins which are transmembrane, they span both sides. And some of these proteins will be extrinsic, which means they're on one layer on the exterior of the cell. So if this was the cytoplasm, change that like that. This was the cytoplasm. This would be the intrinsic area, interior of the cell, and this would be the extrinsic, exterior. So we say that this purple protein here is an intrinsic protein, and this protein here would be an extrinsic protein. Now, extrinsic proteins are proteins that are only in one side of the membrane, one layer or the other, often act as signaling proteins. And what is cell signaling? Well, cell signaling is essentially a way of cells communicating. So when a chemical messenger arrives to the exterior of the cell, and it needs to bind with a particular receptor. Certain cells that are going to be responsive to these chemical messengers, these hormones or these local mediators like histamine and things like that, are going to have specific receptor proteins that are going to cause a response intrinsically within the cell. So cell signaling is just a way of sending messages, and the exterior of the cell has to have receptors in order to respond to that. And it's the same intracellularly. So if we make messages or proteins within the cell, in order to respond to that message, we have to have the correct receptor, which comes in the form of these proteins that are spanning one part of the membrane. So what are transmembrane proteins, and how are they involved in the function of the cell? Well, transmembrane proteins act as channels or carriers. So the difference being, anything that's charged can't cross the membrane. And that's because these phospholipid heads are positive in nature and the tails are negative in nature. And as a result, that means ions that arrive that may want to cross the membrane, let's take a sodium ion, which is positive, will be repelled by opposite charges. So although the tails here may be negative, the head is positive, so therefore the sodium arrives and is repelled by the positive head because of that positive charge. So we take a negative ion, like chloride ions, they might be attracted to the head because it's positive, but then once they reach the interior where the fatty acid tails are, they're going to be repelled away and won't be able to cross. So the only way for these ions to cross the membrane here is to move through a channel protein, which creates an environment where the charge on the fatty acid tails and the phospholipid heads won't have an effect, so they can move through. Now we also need carrier proteins to move molecules which are too large, things like glucose. So because glucose molecules can't fit through the gaps, they're too large, we have to have carrier proteins that change conformation and move those across. So that might be in facilitated diffusion or in active transport. We'll go back to transport in another video. So the membrane has all these proteins, and remember we said the membrane was fluid, so it can move. These proteins are constantly in motion up and down the membrane, and depending on the function of the membrane and what it does, it might have more or less of these proteins involved. So without getting too much into transport, which I'll cover in another video, the final thing we're going to talk about is the permeability of the membrane and how that is affected by its structure. So there are three things that can affect the permeability, which just means how easy things get through when we're saying permeability. And three factors are temperature, saturation of the fatty acid tails, and finally, the level of cholesterol, which is, remember, a lipid. So how that works, if I go back to my diagram of the membrane here, is fatty acid tails themselves can either be, that's this part here, fatty acid tails can either be saturated in nature, which means they look like this, and if we zoomed in on the carbon chain here, it would have carbons with hydrogens at each point on the bonding, meaning it's completely saturated with hydrogen. At every point it can have a hydrogen, a carbon has one. So that gives us a nice long straight chain, and therefore all the phospholipids line up nice and straight next to each other. So this one that I've drawn on the board here seems to have lots of saturated fatty acid tails. However, if we have a tail which is unsaturated, the tail looks like this, which means it's kinked. So it has this structure. So that means the tails won't line up next to each other in a uniform region like they did, in a uniform arrangement like they did with the unsaturated tails, the saturated tails, sorry. So the kink structure creates gaps in between, so that increases permeability. So saturation decreases permeability, and unsaturated tails increase it. So what's going on in terms of the carbon bonding here? So talked about this here in saturated tails. In unsaturated tails, the carbons have double bonds between them. So they have some oxygens where they might normally have a hydrogen, and that structure there creates a kink in the tail. So that's what causes this kink. And that means they don't line up next to each other, and that creates gaps, increasing the permeability. So the temperature then, on the top bullet point there you can see, all right, if the temperature increases, that's going to increase kinetic energy. It's going to mean all the phospholipids are vibrating more and moving around, and because they vibrate more, there's more space in between. But also, the proteins that are present in between the phospholipid bilayer, they will change shape. The tertiary structure will change, and therefore, because they denature, they change, gaps are left where the proteins used to be or have changed shape, and that creates a route for substances, materials to enter the membrane. So that increases permeability. So temperature increasing increases permeability. So that's that and that. Finally, cholesterol. So cholesterol is a lipid, and it exists between the fatty acid tails of the phospholipids. So we'd be looking here at cholesterol. So I like to think of this as a bit like a three-legged race. So if there's a band or a molecule of cholesterol between these fatty acid tails, it sort of clings them together, it wraps them together, pulling them tightly and creating less space for things to move through. So the more cholesterol, the more there is a control of what enters. So more cholesterol equals less permeability. And obviously the converse, if you've got less cholesterol, you've got less binding, that means less permeability, and therefore less movement of substances in and out. So we'll make another video on transport to talk about the differences between those, but that's how the membrane works and how its structure is related to its function.